Lori, are you ready?
There we go. Recording is in progress. Again, thanks for coming out, everyone. It's welcome to Waquoit Bay for those who maybe haven't been here before. I know it's a rough day to be traveling. So thank you, Joan. Like she said, I guess I'll start like quick little thing about myself. I've been working here for about 1.5 years. And before that, I was actually working in Washington, D.C. for the National Geospatial Intelligence Agency in their maritime department. It's been a long circuitous route to and from working in marine sciences, and I'm glad to be back. So, but anyway, on with the show. So I'm going to take you into the SWMP and BayWatchers metaverse. Hopefully, this means people can see things. Great. So no, this is not going to be an hour-long talk on how SWMP and BayWatchers are being implemented into some sort of interactive avatar-based virtual reality world built by rampaging, egomaniac, uber-wealthy people as a new mine for data and profit extraction. No, it's not that. Instead, I'm going to be talking about the sort of universe of metadata and background information that goes into our 2 water quality monitoring programs here at the reserve. I will also get into some of what they can tell us, some case studies of examples of how these programs have been used in the past. So, like I said, we'll start with introducing the system-wide monitoring program. We'll talk a little bit how you all can access SWMP data, and then we'll get into a little bit of what I call maybe complicating that data a little bit, talking about the QA/QC process, some of the strengths of the data set, and then we'll get into a little bit of the case study. Finally, we'll actually approach the Waquoit BayWatchers program, some of whom are here today, which is great. We'll highlight some of the data they have that they've collected over the years, and I'll show you how to access their data as well. And finally, we'll wrap up with a sort of other case study using BayWatchers data. So the system-wide monitoring program. This is the, let's say maybe not the, but a flagship program of the whole National Estuarine Research Reserve system. It is NOAA supported, but then each state agency for every reserve across the US partners some of their funding with a state agency. For us, that's Massachusetts DCR, Department of Conservation and Recreation. So we are NOAA and DCR supported. This support and funding goes to water quality and weather monitoring, salt marsh vegetation surveying, land use and habitat change analysis, and it's all towards the goal of identifying and tracking short-term variability and long-term changes in estuaries. We're going to come back to that phrase: identify and track short-term variability and long-term changes. At Waquoit Bay, we have 5 stations that fall under the SWMP category, 4 of which are water quality sites, one of which some of you may have driven past, is a meteorological tower for our MET data. 3 of our sites, which I'm for now highlighting in red with our fun acronyms, WQB, Waquoit Bay. The next 2 are the site acronyms. So for instance, Charles River would be CR, Vitoxet Point, MP, Sage Lot Pond, SL. Then the final WQ stands for water quality. At each of those sites, we have a data logger that is put in the water. Camera, here you go. This is what it looks like. People, this is what it looks like. So this data logger sits in the water at these stations for 4 to 5 weeks at a time and collects a whole bunch of data. We'll get into it. We have a 4th station here in the southwest corner of Waquoit Bay. This is Minot Yacht Club. I've highlighted it in purple because this station is special for 2 reasons. 1, it is year-round. The other 3 water quality stations, we can really only reliably monitor about April through December. Weather conditions, state of the water prevents us from really going out in January to swap out songs and things like that. So, but at Minot, we can actually monitor year round. It's also special because that is connected to the geostationary operational environmental satellite, GOES. You might hear me say GOES or GOES satellite. That's what I'm talking about. And that satellite communicates with our centralized data management office, which means you can track live data through the CDMO, we'll call it. We'll get into it. It's telemetered. You can get live data. You can also get live data off of our MET station as well, Carriage House. So WQB CH MET. These data loggers that we put in the water, which I will call songs, they are, like I said, take one out, put one in. So they're pretty much continuous. And every 15 minutes, they take readings, whether it's always temperature, salinity, pH, dissolved oxygen, depth, turbidity, even chlorophyll efflorescence. There's another aspect to our swamp water quality monitoring program, and this is the nutrient section. We also, in addition to this sort of continuous 15-minute data, sample every month from our 4 water quality stations. We take water samples in 2 ways. One, with this sort of R2D2 looking contraption we call our ISCO sampler. And this is meant to collect 12 samples along with 12 replicates every 2.5 hours over the course of a full tidal cycle. So about 26.5 hours. We then, of course, also do the good old traditional stick some sampling bottles at the end of a long pole, dip the pole in the water. There's your water sample. We take those samples and send them off to a lab with a University of Massachusetts at Dartmouth, their School of Marine Science and Technology lab in New Bedford. They analyze it for a whole suite of nutrients ranging from various forms of nitrogen to various forms of phosphorus, as well as carbon and silicates. Where do I get all this data, Theo? You're talking about all this data. Where do I get it? Well, there are a few ways to get at it. One, do I dare make this work? Let's see if I can. One way to do it is by going to our homepage. And I'm going to change this screen share here to screen 1. Hopefully that works. So, oh no. Well, regardless, what you can do is go just to that website this is our reserves website, main homepage. What you do is you scroll down to the bottom and find an icon that says WQB CHMET or WQB MHWQ, water quality. You can click on that and get to live data. Another option is, actually, I'm going to stop share here and clear this out and start sharing again. So we won't try going to the links. That's fine. It's okay. The other direct link you can do is the nersdata.org/mobile. This is the SWMP mobile app. It leads you to the exact same place, but the reason I'm bringing it up is because it's part of our centralized data management office's website. You'll hear me refer to them a lot as the CDMO, and I do want to plug them. They're doing a good job of collecting our data and distributing it. So I want everyone to kind of know about these 2 ways to get there. But you can also go to the CDMO website and download their data directly from them. You can select what station you would like to see data from. You can visualize that data right on the site. You can even choose particular time segments of data and download that. No matter how you download from the CDMO website, you will end up with a zip file. And that zip file is going to have a whole ton of information, basically complicating the data that you just received. There's a whole lot of information that goes into it. Why? Because it does take a lot to go from bringing data from your data logger to a table. So how does data go from SOND to table, so to speak? The shortened version is this. You start off with data from your data logger. We download it to our computer, send it off to CDMO. CDMO actually takes a quick look at it, ingests the data into its database and says, is anything wildly off base here? Okay, then yes, no, whatever, they send it back to us. We then, as the year goes by, aggregate and build up all of these deployments and all of those QA/QC documents and go through it parameter by parameter, row by row, and build up an annual document of water quality for each station across the whole year. And then at the end of the year, we send that really intensely QA/QC suite of data back to the CDMO and say, here, take one more look at this before authenticating it and making it official. So this goes through 3 rounds of QA/QC data, and it can be available to people beforehand, but you'll be told that it's provisional data. Only after it goes through its 3 cycles of QA/QC is it officially authenticated. The whole point of this is just basically to try to make this data set both as robust and internally consistent as possible so that we're applying the same standards across the whole year's worth of data. But what does all this mean? It means that a lot of data gets rejected, labeled as suspect. And in general, this finished data set looks a little different than it was when we first downloaded it. So for instance, this is a recent deployment I made at Minot Yacht Club. This is what it looked like. This is what the data from the SOND looked like when I first downloaded it to the computer. The beginning part of it looks great. Only in the last few days of the deployment, the battery died. Cold water, cold environment, batteries tend to lose a lot of power in colder environments than warmer environments. I was lucky, I was able to catch that. Maybe like, it was probably only down for about a day before I caught it and was able to replace it. But what if that SOND had been at a non-telemetered site, you know, at Sage Lot in the middle of summer, and maybe it stopped recording in its 2nd week. Maybe a sensor stopped working and started recording bad data. I wouldn't know until 4 to 5 weeks later after I retrieve it. And then you can end up with data like temperature data from Sage Lot in November 2022, where there's a segment missing. Oops, too bad. And this happens sometimes. It even happens with our nutrient data. We get our nutrient data from sort of 3 times throughout the year. Okay, 3 to 4 times throughout the year, we get it in bunches from our lab. And after we go through and flag it, it looks really messy. It's just spreadsheets full of numbers, yellow, crazy everywhere, okay? And some of these flags and codes and comments are meant to provide more perspective for people who are using the data. For instance, you can look at it and say, okay, in one of the columns here, we're seeing a flag that says -4. Well, that means that the lab can't reliably tell if that value they got was a real value or a blank. Below a particular level, they can't tell whether it's a real result or just plain old deionized water, okay? And so what they do, we have to find a way to alert people and say, here's a number, approach it with caution, okay? The other thing that can happen, let's say with our nutrient data, is maybe your sampling is going great all the way through 2022 until June of 2022, when your fluorometer breaks and you can't measure chlorophyll A concentrations that month. That's just a hypothetical. I'm not saying that actually happened. Am I convincing? So aside from these sort of logistical issues that can happen with swamp data set, there are some other sort of limitations. And the biggest one is that we have 4 stations over the whole lot of Waquoit Bay and its watershed. This map here, you can see these 4 stars are the locations of our water quality sites. It seems like we have the bay decently well covered, although maybe, who knows, there could be some heterogeneity within the bay. Maybe the southern part of the bay near the cut where it's more connected to Vineyard Sound would be different than the central part of the bay, which may even be different than the northern part of the bay. We have several tributaries which are not really monitored, and it'd be really nice to have some sort of data that tells us, hey, this is what the water quality is, not just in the bay, but the waters that feed into the bay. Our swamp data set doesn't really tell us that right now. And then there are the issues of monthly data. I said that we go out monthly with our nutrient data and collect samples. Well, 12 data points over the course of a whole year is not the best time resolution if you want to see patterns over the course of a few months. Some patterns might be hidden from you. But then there's the other sort of philosophical question of, oh, one data point in March, one measurement in March. Well, in March of 2022, we sampled in March 8th. March 2021, we sampled probably on like the 16th or 17th. A lot could have happened. It's not like that March 8th sample is totally representative of all of March. It's not as if processes in the water that are happening on March 8th, chlorophyll isn't saying, oh, hey, it's March 8th. Let's do the March 8th thing today, okay? We have to be really careful about imposing a monthly reference frame onto processes that don't necessarily follow that. Not to mention this 12 data points per year, a little bit of a low time resolution. But this is me really, really, really unfairly beating up on our swamp data set, okay? Who really cares if we're missing some data every once in a while? We have 20 to 25 years of data or more at some of these sites. For all of those years, the sample methods have been consistent. We've been applying consistent and really standardized, rigorous QA/QC processes to all of them. And on some level, who cares about all these having the perfect timescales for all the data? We have continuous water quality data, which can work at many different scales. We have monthly nutrient data, which can really work for, maybe it doesn't work really well for tracking short-term variability, but it does a really good job of tracking long-term change over decades. And then we also have our diel nutrient samples, which can really help us with variability over a 24-hour period. So you can use this data to telescope down and say, here's water quality over a long period of time. Here's nutrient data over a much shorter period of time. On some level, we're running up against the Kurt-Gödel incompleteness theorem here. We can't have a data set or a frame of reference that's both entirely complete and internally consistent, okay? You can't have something that is both completely perfect and perfectly complete, okay? And swamp can't be that, but it is more than good enough. And there have been researchers who've been using swamp data for years. And over the past few decades, it's been the swamp data set that's really helped deliver a message that the most important water quality issues in Waquoit Bay are eutrophication and climate change. For people who have not heard the term eutrophication before, it's a long word, and it basically means the enrichment of a water body through increased nutrient inputs. I will skip over global warming because we should all know what that is. I will say a really, really, really great example that I'm going to go into now of swamp data being used is from a 2023 paper by Matt Long and Jordan Mora, who used to work here. Jordan Mora, she used to work here. Matt Long has talked about this. He talked about it last year at this very talk. Jordan has talked a lot about this too. They are both much more qualified to talk about their results and their work and their paper than I am. But I'll give you the quick hits as just an example of how SWMP is being used by researchers. So they specifically looked for some of these patterns at the 3 sites within the bay: Charles River, the Toxic Point, and Sage Lot. They kind of found that Minot behaved a little differently because it's more directly connected to Vineyard Sound. But at those 3 sites within the bay, they found strong evidence for an increase in water temperature. Using the SWMP dataset, they also found that there's a decrease in dissolved oxygen. This is between 2000 and 2020. This would be expected because warm water can hold less dissolved oxygen. These are just sort of basic principles. But they also found a correlated increase in the acidity, so a drop in pH in the same time period. And in that same time period, they also found, using the SWMP data set, between 2006 and 2020, an increase in total nitrogen. Along with that total increase in total nitrogen came an increase in chlorophyll A. Chlorophyll A is a photosynthetic pigment used by various types of macroalgae and microalgae in the water. It helps them photosynthesize, create sugars, and then they use nitrogen. Then those same organisms use nitrogen to grow. But this is an issue because Waquoit Bay has traditionally been an ecosystem dominated by eelgrass. That eelgrass has been, over the decades, slowly wasting away and disappearing. And they found over the course of just the 2000 to 2020, an increase, like a correlated increase between a loss of eelgrass over that time and an increase in chlorophyll A. What they're saying is that this is sort of a shift in the type of ecosystem that we have in the bay, a restructuring from one that's based around eelgrass to now one that is based around phytoplankton and algae floating in the water column. This is a significant change in an ecosystem that's being tracked and sort of elucidated by these researchers using SWMP data. That's the end of our case study for SWMP. But let's go into Waquoit BayWatchers. So as much as we love SWMP and we love how it's this sort of flagship program for the National Estuarine Research Reserve System, Waquoit BayWatchers is something different. This is a volunteer-based water quality monitoring program. It started in 1993, so they have over 30 years of sampling and data. Let's compare that to SWMP for a second. SWMP, as a program, started in 1995. We were already operational at Waquoit Bay by 1995, but our oldest continuously run SWMP station started in 1998. So we actually have a significant amount of BayWatchers data that predates the SWMP program. Their goals: to track trends and changes over time, document seasonal variation, maintain a reliable stream of information for researchers and decision makers, and to increase public awareness, education, and involvement. BayWatchers, this is a really great slide, but it's also kind of confusing. BayWatchers has 10 sites they sample from, and we sort of split them into 3 different sections. We have the east side and the central side that I'll talk about together because those 2 groups access their sites via boats or seasonal piers and docks. So they can only sample pretty much May through October. The west side team, they can sample year round. And during the time span between May and October, sampling happens bi-monthly, twice a month. The west side team will also sample twice a month during that time, but then for the rest of the year, between November and April, they're sampling once a month. So we have data year round now. These volunteers, some of which are here tonight, they go out usually early in the morning around a low tide to be consistent with our swamp data collection methods. And they go out and collect surface and depth water samples. They do so with the same hole and sampling bottles that we use for swamp. They use a YSI handheld device, which is pictured here at the top, to record field data. And then they take those samples back into our lab and filter them. They filter them for chlorophyll. They filter them for dissolved nutrients. And then they take some unadulterated, unfiltered water. They take all those things, put them in a freezer, and we have folks from the Center for Coastal Studies in Provincetown come and pick them up. They perform the laboratory analysis and some QC on the data before sending it back to us. And I take a look through it. And then I also record with that laboratory data, our field data. So all of the chlorophyll data, all of the nitrogen, phosphorus, carbon, and silicate data that we get back from the Center for Coastal Studies, I match up with that sample's temperature, salinity, dissolved oxygen data. So we have matching data across for the whole year. BayWatchers data, all about it. Let's highlight it. So one of the strengths of it is that we have 10 sites. Yes, more than 4. So higher spatial resolution than swamp. And it covers our upstream areas that we weren't able to get with swamp. This is one of the ways I personally use the data set. Is to sort of get a little picture for what the water quality is, the water going into the bay. So for instance, if we want to look at nitrogen, nitrogen is a nutrient in the water that helps phytoplankton grow, okay? It's linked with biological activity, okay? And maybe we want to know what are the various forms of nitrogen in the water. So this is actually taken at Site 5, which is the Waquoit River, which is the probably furthest northeast site in the bay here. Okay, the Waquoit River, we will also call it the Municus River. And generally what we see is sort of a spread. There is some nitrogen that's particular organic nitrogen. There's some dissolved organic nitrogen. There's very little ammonia, but there's a decent amount of the NOx. NOx are nitrate and nitrites, okay? If we know a little bit about nitrogen and estuaries, one of the sort of basic things is if the water is moving, it's going to be oxygenated. In an oxygenated environment, like maybe a river that's running, we get a lot of bacterial activity that converts ammonium to nitrate. So it kind of makes sense that in this environment, we wouldn't see a whole lot of ammonium for most of the year because as we are sampling it, it's actually being taken up and converted into nitrates. But we can also compare this and say, okay, this is a picture, a snapshot of our water quality in an upstream area. What happens when it gets flushed out to the bay? Well, let's compare it to one of our sites in the central part of the bay, site 9. Okay, by the time we get to site 9, all of those nitrates, not all, but a lot of those nitrates are gone. And our water sample is dominated mostly by particulate organic nitrogen, dissolved organic nitrogen, and again, a little bit of ammonia. And this is because I would interpret this as saying we're further away from our groundwater sources that are bringing nitrates into the water. We're further away from our runoff sources that are bringing nitrates to the water. This same water has had time to go out into a larger body and get diluted. And there's also been time for other organisms that use nitrates to uptake them and use them to incorporate them into their bodies and help them grow. This makes sense. But let's go even a step further and say, we have a swamp station right next to this BayWatcher station. They're basically in the same spot. Let's corroborate, let's look at them together, and can one corroborate the other? We'll feel really good about our data sets if they're both saying the same thing, right? And so this is the sort of distribution forms of nitrogen at basically the very same spot. And this is the swamp data set. It's a little weird to look at, but the message I interpret is that it's more or less the same. We see a whole lot of particulate organic nitrogen. We see a whole lot of dissolved organic nitrogen, and we see very little nitrate. And again, a decent amount of ammonia. So again, one of the kind of limitations of BayWatchers data is we're still doing monthly sampling. We still have that kind of low time resolution, but we can still see general trends. I think a really good example of this actually that I kind of talk about a lot is air temperature and water temperature. And this is a figure of air temperature in Massachusetts, recorded in Blue Hills since the mid-19th century. It's going steadily up. I'm going to hypothesize that if air temperature is going up, and it's going up steadily for decades and decades and decades, we'll probably also see an increase in water temperature. That's probably pretty fair, right? Maybe? Okay, great, some nods. And indeed, no matter both our seasonally sampled sites and our sort of like whole year sampled sites, we do see a warming trend in surface waters of Waquoit Bay, thanks to BayWatchers. Now, we don't have data going back to the mid-19th century, but you know, I think mid-90s is okay, is a pretty robust data set too. That's a pretty good timeline. If you want to access BayWatchers data, this is one of my favorite parts. If you want to access BayWatchers data, the best and actually only way to do so is through the Provincetown Center for Coastal Studies. They have a really, really, really great water quality program website. And it's I find it pretty easy to use. This is what it looks like. You can zoom into Waquoit Bay and then basically select, I think I selected my favorite site, Waquoit River. And you can look and see the general trends. This is what temperature did in 2022. The line tells you this is the actual data. The shaded region is all the other previously recorded data. So you can see how it even compares to past years. You can scroll down and actually kind of select various parameters that you want to see, that you want to look at and compare them against each other. I selected here chlorophyll and nitrate and ammonium. And the reason I did this is because I want to kind of talk a little bit about something else. As chlorophyll goes up and spikes, let's say in June and July of 2020, we see the orange line representing the nitrates dive down. And that occurs again during the late spring or early summer of 2021. Chlorophyll goes up, nitrate goes down. And this is us actually probably being, I interpret this as us being able to see on some level. You could even think of it as sort of like, I don't want to say the bay breathing, but this is, you see chlorophyll go up. This means you have much more active biologic activity. As they are becoming more active, they're incorporating nitrates from the water, and they're taking it up, and all that nitrate in the water, that concentration goes right down. Pretty powerful stuff that you can see, and it's a great tool from the Center of Coastal Studies. The other place you can find Waquoit, so let's say a case study for Waquoit Bay's BayWatcher data, was the Cape Cod Commission. You can't find Waquoit Bay, you can't find BayWatchers data on the Cape Cod Commission's website, but they are cited in the Waquoit Bay 2020 TMDL. TMDL stands for Total Maximum Daily Load. And this is part of a long decades-long research done by the Massachusetts Estuaries Project in collaboration with the Massachusetts DEP to quantify how much of certain nutrients make it into various water bodies, and then try to identify what would be a more healthy target level of these nutrients. So almost think of it as sort of a diet plan for these bays and waters across Massachusetts. The sites that, the figure on the left here shows sampling locations around Waquoit Bay that went into making the TMDL. And a lot of those, the starred ones the ones I put stars next to, and those are sites that I've also highlighted in the table on the right. Those are BayWatchers locations. So BayWatchers locations was aggregated by, BayWatchers data from all these sites was aggregated by the Cape Cod Commission and used by Massachusetts Estuaries Project and DEP to create a TMDL for Waquoit Bay. And here's a table. We're going to go back to my favorite site, Waquoit River. So they're saying that, according to BayWatchers data, they're saying that the Waquoit River delivers about 20.51 kilograms per day of total nitrogen. And that a target load for it should be 13.47 kilograms per day. It's a pretty steep reduction. TMDLs are mandates. Progress towards these goals is being tracked. We're required to meet these TMDLs. If we don't meet them, the state has to spend more money and more time and more effort towards getting to those goals. So let's perform some kind of cursory check on those goals. We can use some data from BayWatchers to see how we're doing. So that's what I did. I said, okay, here's our total nitrogen concentrations at the Waquoit River throughout all of 2022. I then also used stream flow discharge data from a USGS stream gauge that's in the Waquoit River right nearby. And I was able to convert micromolar concentration to kilograms per day and discover that in lots of 2022, we were over, for every sampling event in 2022, we were over the TMDL threshold in the Waquoit River for Waquoit Bay. So at every sampling event during 2022, sometimes we were over the TMDL limit, sometimes significantly over. I hate to end on a bummer, but the bummer, really, I do want to stress that this is a kind of success story for the use of data and the various ways that volunteer water quality monitoring programs can work. They're successful. They can really contribute a lot to existing well-run data sets. They can be used for, state and federal coastal management, resource management policies and issues. And so to me, that's not a bummer. That's a real positive. So on that note, I do want to thank all the BayWatchers volunteers. I want to thank the Friends of Waquoit Bay Reserve for finding all these volunteers. I want to thank NOAA and Massachusetts DCR for their funding. The lab people at Center for Coastal Studies, the Cape Cod Commission, our labs at UMass Dartmouth for providing us all this great analysis. And that's what I got. Questions?
Very good and detailed, but understandable and clear, I thought. And so yeah, we're going to try to do, so we have a bunch of people on the webinar, as well as people in the room here. So we can start in the room, and then Laura, you're good for watching the chat box, right?
Absolutely. I've got one question in the queue.
Okay. Well, we'll start here with you, sir.
Theo, I just had a question on the swamp data logs.
Yes.
Data logs. You said you only have 1 out of 4 that you can monitor remotely.
Live, yes.
Yeah. And why is that? We just don't have the equipment, or it's not working?
It's tough to get. I mean, right now, what we have, it's kind of hard to. I can show you. It's a little hard to see over the camera, but basically, these data loggers have, a connection for like a flying lead cable, and that cable connects to an actual satellite transmitter and signal data input, sort of organizes all that data. There's a program for it. So we're talking, you need power, you need actual physical infrastructure nearby on which to place an antenna. It's not really feasible out in the middle of the bay. So that's the biggest
money come into it as well.
Money's a big thing too. If we had money that could put in some infrastructure out in the bay, we might think about it. But even then, yeah.
Okay, we'll take one from the chat box, and then we'll come back. Just give them a chance.
I'll wait.
All right. From Emma, very interesting talk, Theo. Well done. What is the purpose of testing for silicates?
That's a really good question. I'm going to pass this off actually to Megan, who's here. But no, that's a really good question.
Probably diatoms.
Yeah.
For the productivity. And
yeah.
Wally's lab really likes it. So it's not required.
Yeah.
But we do it anyway.
So in case you couldn't hear that, of the types of phytoplankton we have in the water, a family of them, which are very, very numerous, are silica-based, called diatoms. So that could be a way of tracking them. And we have a lab that we work with, a university lab that we work with, who sometimes likes to see that data. So,
hey, Alex had her hand up, and then go next after Alex.
On the last slide, the TMVL spike.
Yes.
Lucas, do you know if that correlates with at the same time when there's that scum all over the river?
I'm hesitant to correlate it to anything, considering it's 1 year's worth of data. But this is in midsummer, when we do happen to get a lot of algal blooms. And so lots of times algal blooms can be responsible for sort of scum that you see on the surface of the water. I don't want to give a definitive answer, but.
So my question was relative to population density, especially in this area. Mashpee, I don't know what the actual percent increase is now after COVID, but it's still significant. It's, let's say 50% for the record. And when I look at that, I can only wonder what the human impact is in terms of overuse of septic systems, things of that sort. It's time to mow my lawn and put some fertilizer, you know, all of that stuff.
So, I would say that the primary reason why we have elevated nutrient levels in the bay is because of human activity and the way humans have decided to manage the land on Cape Cod. Yes.
That wasn't meant to be a softball either.
Yeah.
But you can name a couple ways.
You can name, yeah, and it's related to septic systems, the over-reliance on septic systems. It's related to over-reliance on agricultural fertilizers. Actually, I don't have this slide right here, but it's interesting to compare this site with swamp data from a site in the Charles River. The site in the Waquoit River is a bit more forested, not as developed as the site near the Charles River. And particularly you can kind of, I don't know, you can infer that the differences could be to the different types of land use in the area, both historic and current.
Yeah,
So
you would certainly need much more data.
You need much more data and time, but
there have been studies like that. Lori, anything in the chat box?
I have one more. Theo, do you check on the sonde at all during the 4 to 5 week deployments? And how do you stop the equipment from getting fouled during that time?
I think that's a great question. I do check the live data almost daily if I can, maybe not on the weekends, but certainly most days during the week. And if I think that there's fouling, I'll go and do a quick check and clean things off. But otherwise, you get pretty good flow of water through the sonde, and at that site, there isn't a whole lot of still water happening. So you'd be surprised, there isn't as much like fouling community growth on there. Lots of times we do see lots of crabs though. So.
Thank you.
More questions in the room here? Yep.
Who's the biggest user of all this data? You named like a few of the
man.
that use it.
I wish I knew.
Yeah.
So when you go to the CDMO website, you can choose all the data that you want, and they send you a zip file. They send you a zip file with your email. So they ask you for your email, they ask you for your name, they ask if you're associated with a research institution, they ask you some questions. I would love to see who's using our data, but I haven't checked on that yet. So
Okay.
That's not available to me.
I just curious.
That's a great question. I'm so glad you asked that. So.
You more in the chat box, Laura?
I don't have any pending questions.
Okay, you more in the room? All right, well, thank you so much, Theo. And this is the last in the series for this year. I'm sure we'll have more next year. And also we have a lot of events coming up in April and May. So check out our website, or if you're here, you can take some flyers. So thanks for coming, everybody.
Thanks for coming.